Hi, on this video, I'll be teaching muscle tissues. Um, if you had missed my previous videos, please go ahead and watch some of those chapters. They'll be helpful to understand this chapter as well. Now, muscle tissue, it's what we'll be studying here is we'll be looking at the anatomy of the muscles. I will also be looking at the way the muscles affect our movement, how muscle contract and react and relax. All right. Now, the study of muscle by itself, which is what we want to do now, we call it myology. So anytime you say myo is muscle. When you contract your muscle and you cause your muscle to perform an action, all we are doing at that point is that you are converting a chemical energy, the energy from the food you have, you are converting it to mechanical energy. All right. Now, when you see a skeletal muscle, it's an organ by itself. And so it's going to consist of cells, right? So inside the muscle, you have muscle fibers, which we call the muscle cells. Right. So when you hear myofibers, muscle fibers, they are all muscle cells. Um, so myofiber is the same thing as muscle cell. Fiber is muscle cell, so these are muscle cells. All right. Then we have some connective tissue wrappers that surround them. We have blood vessels and nerves. Now, the ability of a muscle to be able to get influenced by stimulus is what we call excitability. Is the ability of a muscle to respond to stimulus, to be influenced by stimulus, to be activated by stimulus, for an action to be generated by stimulus. We call that excitability. Conductivity is the ability of that muscle that has received the stimulus to be able to spread the stimulus throughout the membrane. All right. At that time, I'll be talking about action potential, which we'll be discussing later. So when a stimulus generates an action, a potential for an action, that stimulus, the signal must be able to propagate and spread across the membrane of the muscles. We'll be talking about that. Now, contractility is the fact that a muscle can shrink, it can shorten. That's contraction. And the ability of a muscle to extend again, to stretch, is extensibility. The ability of a muscle to return back to its initial length after stretching is what we call elasticity. All right. Now, these are the general functions that muscles perform. Muscles are involved in movement. We are able to move our body because of muscles. There are three units that are involved in movement: muscle, joint, and bones. Now, the ability to maintain posture. Now, our posture is due to the reinforcement of muscles. So you see that for you to sit, it is a combination of the muscles of the back and the abdominal muscles. If the abdominal muscles are weak, just like what we see in children with cerebral palsy, you will notice that those children have weak abdominal muscles, and because of that, they are not able to sit. So before you can sit, you can maintain that posture. It must be the balancing of the agonist and the antagonist, which I'll be talking about in the next video when I'm talking about synergies, agonists, and antagonists, which are the functions of muscles. Respiration, obviously, you have the cardiac, you have the diaphragm, right? So for inspiration, we have the you have the muscles, the diaphragm. The diaphragm is a muscle, and it's involved in inspiration. The external intercostal muscles, they are all involved in inspiration. Uh, and so if there's respiration, right? If those muscles don't contract, respiration does not happen, right? Now we have our body heat, our muscles generate body heat, communication. As I'm talking now, muscles are involved in communication. You do facial muscles to communicate. We do signals, all of those. Constriction of organs. Organs opened by sphincters, right? So when you talk about the pyloric sphincter in the stomach, the cardiac sphincter in the stomach, the ana recta, the sphincter, all of those sphincters open by contraction and relaxation of muscles. Now, and also movement of molecules through the body, the lymph, the blood, urine, air, food, they move as muscles contract and relax. Now, there are three kinds of muscles that I'll be discussing. The first one is the skeletal muscle, cardiac muscle, and smooth muscles. Now, the key thing you want to look at is this: where do you find each of them? Where can they be found? The first is skeletal muscles. You see skeletal muscles more like you see it everywhere in your body. There, you see it all everywhere you have bone. You see it around the skin. You see it around the fascia, the membrane below the skin. Under the microscope, the skeletal muscles appear striated. And what we mean by striation is the arrangement of the contractile protein, which are actin and myosin. Actin and myosin, those are the contractile protein. It is the way these proteins are arranged that we call striation. So the actin is what forms the dark band. The myosin is what forms the light. The actin forms the light band. The myosin forms the dark band. That is what we call striation. All right. They are made up of. They consist of multiple nuclei on the surface of the cell, and that should tell you that is reasonable because there's a lot of activities that happen in the skeletal muscles, right? So it carry out rigorous activities. So it needs nucleus because a lot of nucleus simply means that it's going to generate more protein, and more proteins are needed for all of those functions, right? Now the voluntary the contraction and relaxation are controlled by voluntary means. What that means is that it's controlled by the central nervous system. So skeletal muscles are controlled by the somatic nervous system. Somatic nervous system, SNS, somatic nervous system. All right. Now, some skeletal muscles contribute to involuntary reflex behavior, just like what I talked about when talk about breathing, for instance. Now, even though breathing it's done by cardiac muscles, but skeletal muscles play a role in that, right? We talked about the diaphragm, they are involved in breathing. All right. Now, for cardiac muscles, you see them around the heart. That is key thing. Where do you find them? You see them around the heart. All right. They have a single nucleus, unlike the skeletal muscles that have multiple nuclei that are located on the surface. These cardiac muscles have single nuclei that are centrally located. And so we also say they are striated. And striation simply means what you can see here is a pattern of arrangement of the contractile proteins, which are actin and myosin protein. I'll be talking about that later. They are controlled by autonomic nervous system. So I call it automatic. Actually, it's autonomic nervous system, ANS, autonomic. All right, here is autonomic. Autonomic nervous system. Autonomic nervous system. That's a part of the nervous system that controls. Now, you want to think about it. Somatic nervous system controls voluntary actions. Autonomic nervous system control autonomic actions, involuntary actions. Does that make sense? All right. Now, so they have the autoneurodymic contraction, which is what you feel lump dump, lump dump, lump dump, and it's because of the way the cardiac muscles are modified. The heart has pacemakers, right? Now, each skeletal muscles, each cardiac muscles are separated by what we call um, intercalated disc. And I was telling my student in class that when you look at the vertebral bones, right? If you go back to the chapter, the vertebral bone, you will notice that each vertebrate is separated by intervertebral disc. That means there's a disc between the two vertebrae. The same thing here, between each cell, there's intercalated disc. So when you see one intercalated disc and the next, you know that that is a cell in between here. So like this now, this is an intercalated disc right here. And so there should be one somewhere around here as well. That will be a cell, all right? So a cell consists of a nucleus and there will be one intercalated disc. There will be intercalated disc dividing each of the cells, all right? They are like cell junctions that are made up of desmosome and gap junctions, meaning that they are able to allow molecules to travel, to transfer in between them. Now, you know that the purpose of intercalated disc is simply to keep the cells together to be able to communicate because they are cell junctions. So that when you say that cells are group of when you say tissues are a group of cells that works together to perform a function, you want to think that way, that for the cells to be able to work together to perform function, they have to have a means of communication. The means of communication between cells of the heart is what we call intercalated disc. Now, the third one are the skeletal muscles. The smooth muscles. The smooth muscles, they are called smooth because they don't have the pattern that is made by contractile proteins on them. They don't have pattern. So that is why they are smooth, meaning that they don't have striations. All right. They are involuntarily controlled. Again, that means they are controlled by the autonomic nervous system that does automatic actions, right? Now, you see them in the wall of hollow organs. So you think about blood vessels. Hollow organs simply means organs that have holes in between them. Think about it that way, right? So you see them in blood vessels. You see them around the eye. You see them around the glands and around the skin as well. They have single centrally located nucleus. So smooth muscles and cardiac muscles have single centrally located nucleus. Uh, Skeletal muscles have nuclei, meaning that they have more than one nucleus, and it's located on the peripheral, on the surface. Now, each skeletal muscles have, they also make what we call the fascias, and the fascia are simply muscles that are below the skin. All right. So there is one we call superficial fascia and deep fascia. This is simply based on the arrangement, the way they are located. All right. The superficial fascia is made up of areolar connective tissues, and it is deep. It's fat and fat that are deep to the dermis. You see that? So it's deep to the dermis. It's what we call the subcutaneous layer or the hypodermis. That is a superficial fascia. Now, deep to that fascia is another one, and that is what we call the deep fascia. So that means superficial fascia is above, deep fascia is below, right? So they are dense irregular connective tissues wrapped around the muscles and they line the body walls, and also you see them in the upper limb and lower limb. Now, let's look at what a muscle looks like. Now, we'll begin to talk about terminologies. What is key about this chapter is that you want to get familiar with the terminologies so that you can understand it very well. I'm still going to do a follow-up video later when I will likely do this on the board and just record it and upload it, okay? Because I have the limitation of drawing with this PowerPoint. Now, when you see, the first thing I want to tell you is that I told you earlier that muscle fibers are the same thing as muscle cells. You remember? So these are the same thing as muscle cells, all right? And they are also the same thing as myofiber. These are the same thing as myofibers. Now, So when you see muscle cell is the same thing as muscle fiber, it's the same thing as muscle cell, it's the same thing as myofibers. Now, when you look at this, these are muscle cells. So if you count here now, inside there we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So inside this bundle we have about 9 muscle cells. Now, so inside this circle alone, we have about 9 muscle cells. So there are 9 cells inside this place. All right. Now, 9 cells are there. Now, what covers each of the cell? So think about it. There is a muscle cell like this. This is a muscle fiber. It is covered by something, and that is called endomysium. This is another muscle fiber. This is another muscle fiber. Another muscle fiber. Now, the same thing I'm talking about is what you see here. When you come here, this here is a muscle fiber. Now, this is a muscle fiber. There are a lot of them here, right? And one of them is what comes out here, all right? Now, this is a muscle fiber. That means what covers this muscle fiber, what is covering this muscle fiber is what we call endomysium. Does that make sense? Now, when you look at this, there's now another bundle because there's a bundle of muscle fiber, all right? This is another bundle of muscle fiber. This is another bundle. So this is a bundle of muscle fiber. This is another bundle of muscle fiber. This is another bundle of muscle fiber. This is another bundle of muscle fiber, meaning that a group of muscle fiber. The group of muscle fibers are surrounded by what we call perimysium. All right. So you see that perimysium is here. So perimysium is surrounding the muscle fibers. See that? Perimysium is surrounding the muscle fiber. And when you look at it, it's a dense connective tissue. Now, when you have a group of muscle fiber, now these are muscle fibers, right? Each of these muscle fibers, when you have a group that form a bundle together, we call this group fascicle. So in essence, you are saying that each fascicle is surrounded by perimysium. All right, so when you see the myofibers, they are surrounded by endomysium. A group of myofibers form what we call fascicle. So this is a fascicle. And each of the fascicle, you see the fascicles are surrounded by that dense connective tissue there, which we call perimysium. All right. Now, a group of fascicles, so this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. This group of fascicles is what makes a muscle. And that is surrounded by epimysium. Epimysium. It's a dense connective tissue as well. Now, you see, it's a dense connective tissue surrounding a whole muscle, meaning that many fascicles, it allows muscle contraction with stability. All right, let's go. Now, tendons are simply the connector between muscles and bone. Or so you see, now when you have a tendon that is flat, we call it aponeurosis. Now, nerve supply, so the muscles and the blood supply. General rule is that blood supply to the muscle is in this format. Each of your skeletal muscles receive blood from one artery and they will be drained by 2 veins. So one artery supplies, two veins drains. Also, for nerves, a single nerve supplies each skeletal muscles with information from the brain. So information from a single nerve is going to supply each muscle nerve fiber, each muscle fibers, each skeletal muscles. Now think of it this way. A motor neuron is a neuron that we also call afferent neuron, right? Afferent neuron. Afferent. Afferent, efferent. So they are neurons that take information to the nervous, to the muscles. All right. So we call them motor neuron. Motor neuron. So if you are on the next chapters, I'll be talking about neurons, right? But then we have sensory neurons and motor neurons. Sensory neurons are the neurons that take information into the central nervous system. A motor neuron takes information out of the nervous system. All right, so don't forget that motor neuron takes information out of the nervous system, and sensory neuron takes information into the nervous system. All right, now, so those are the neurons that innervate or supply the skeletal muscles are generally called motor neurons. All right, so here I will start to look at the anatomy of the um, muscles. I want to discuss that. I want to pay very close attention. I'm looking at the anatomy of the skeletal muscles. I'm going to look at the anatomy of the neuromuscular junction, meaning that junction between the neuron and the muscles, the activities that happens there, the activities that happen in the muscle fibers, and how relaxation happens. All right. So they are very interesting sections that we will be looking at now. So the first thing I want you to see is that first is the way the muscle fibers or the way the muscle cells are arranged or they are built, they allow information that comes from the brain or from the nervous system to travel through them, and it will cause effect. Now, first thing I want you to see here is that there is a portion on the portion on the skeletal muscles where the neuron is going to relay the message that is coming from it is called the motor end plate. But before I go into that, I think I should explain this first. Now, when we say the muscle is going to contract or relax, think about it. What makes it to contract? What makes it to relax? Why is it that somebody that has stroke cannot contract their muscles? Why is it that somebody that has stroke cannot contract and relax their muscle, even though they still have the muscle? Why is it that a child that have cerebral palsy cannot contract the muscles and relax it, even though the muscles are there? Obviously, over time, lack of usage is going to cause the muscle tone to reduce. So, but the muscles may be there and not be able to contract and relax. Now, that tells me that what caused the muscle to relax and contract is not the muscle by itself. There's something that controls it, and that is the brain. So the central nervous system is still the controller that decide when the muscle is going to contract and when the muscle is going to relax. So that means when you receive a stimulus, let's say you saw a big fire now, and naturally you should run, but your leg, your lower limb, your upper limb does not decide to run. What determines when you are going to run is that there is a message that comes from the brain and tells you to run. Now, if the part of the brain that dictates that message is destroyed, is dead, or is damaged, which is what happened in stroke patient, then the muscle can be there and yet not contract. So a stroke patient is going to see fire and not be able to run because there's nothing to tell the brain to tell the muscles to contract. So now what we'll be looking at is we'll be studying the anatomies first, understanding the structures, but what we are looking at is this: how does the message come from the brain to the nerve, to the muscles? How does the message travel from the brain and say, now brain looks at the message, interpret it and say, now we need the muscles to contract. How does that get to the brain, to the muscles, for the muscles to do the dictate or the beat of the brain? So that's what we want to look at in a very quick manner. Now, the first thing I want you to see here, there are some terminologies you just need to get used to. There are things you know before, but they have different names here. Now, sarcolemma is the same thing as plasma membrane or cell membrane. So when you look at this, for instance, now, there is a membrane that covers this whole structure. This structure, this here is a sarcolemma. You see that? This membrane that covers the whole of the structure is a cell membrane or plasma lemma, because it has to sarcolemma, because it has to do with muscles now. Now, the sarcolemma have a structure, it has some structures in it. And the first thing I will talk about is the T-tubule. Now, here is the T-tubule means transverse tubule. You see that? Transverse tubule. And here's the T-tubule. Is a yellow stuff you see here. That's it. And it's transversely located. You know, it's transverse. Think about if this is the muscle, the T-tubule is transverse on the side, right? So this is a transverse structure. That's why we call it transverse tubule, and it's a tube. If it's a tube and it's in the body, then it will be used to pump something. I'll be talking about that later. Now, there is another structure that is closer to the tube, and it is this blue stuff here, this blue structure. We call this structure terminal cistern. Terminal cistern. Now, you need to understand that cistern or cistern, it simply means a container. There's something that is inside this container, and I'll be talking about that later. So think about it. The way I pictured it for my student in class is, think about having a bottle and a straw. The straw is the transverse tubule, and the bottle is the terminal cistern. So now, if you look at this, you will see that this is one terminal cistern. This is another terminal cistern on this side, and this is one transverse tubule. All right. So this is a unit, a terminal cistern, a tubule, and a terminal cistern. We call that triad. We call that triad. You see that there? So triad means a terminal cistern, a tubule, and a terminal cistern. So two terminal cistern and a tubule. All right. So the T-tubule are indentations of the cell membrane that allow action potential of the myofiber to pass to all part of the cell. I will tell you, I told you the T-tubule is a tube, so it's like a straw which helps to send information out. It helps to propagate information, and I will show that to you how it does that. Now, the myofibrils, like I told you earlier, the myofibrils are simply actin and myosin. Actin and myosin. So they are contractile protein, and contractile proteins are actin and myosin. All right, and myosin. Those are what we call contractile protein. Now, these packages of protein take up much of the interior of the cell. That's that. Now, myofilament. Now, think about it. What is a filament? Filament, when you look at a filament or something that have a filamentous appearance, it has strands, it has lines, right? So when we say filament, we are simply mean this actin is thin. Even the name actin sounds thin, right? The actin is thin and the myosin is thick. So the actin makes a thin filament when they are arranged as fibers, and the myosin makes the thick filament. So if you look at this, you see this here, this one is a thin filament, and here the purple or the blue, the purple rather, is a thick filament. You see that? So that is thin and thick filament. Now, sarcoplasm simply means cytoplasm, but because it is not cell, it is muscle. It is the sarcoplasm, or the plasm of the muscle, right? Is cytoplasm of the cell. It contains large amounts of glycogen. So inside the cytoplasm, which is called sarcoplasm, you have a lot of glycogen there. The glycogen is going to be a source of glucose because when it's broken down, supplies glucose. Then you have myoglobin, which helps to carry oxygen. You see that? All right. Now, the next thing I'm going to show you now are the sarcoplasmic reticulum. When you say the sarcoplasmic reticulum, you should think of endoplasmic reticulum. All right? Think of endoplasmic reticulum. All right? Think of endoplasmic reticulum. Now, the sarcoplasmic reticulum is actually what hosts the terminal cistern and the transverse tubule that I told you the other time. So all of this structure here, where you see them, is actually the sarcoplasmic reticulum. So think of this as sarcoplasmic reticulum, and then this is the terminal cisternae, this is the terminal cisternae, and this is the T-tubule. And the T-tubule and the terminal cistern are parallel to each other the way they are arranged. So you see that the sarcoplasmic reticulum is similar to the smooth endoplasmic reticulum, right? It encircles each myofiber, muscle cells, and when myofibers are relaxed, they store calcium ion. So there's calcium ion inside the sarcoplasmic reticulum, which is actually the content of the terminal cistern. So the terminal cistern is part of the SR, sarcoplasmic reticulum, that lie parallel to the T-tubule. Two terminal cisterns and a T-tubule makes a triad. So think about it, have a picture of it. A terminal cistern is a container and it's part of the content of this sarcoplasmic reticulum. The terminal cistern have a transverse tubule, which is like a straw inside the container to pump out calcium ion. All right. So again, what does terminal cistern pump out? It's able to pump out. What of the transverse tubule pumps out? It's able to pump out calcium ion when it's needed. I'll be talking about it. Calcium ion is very important. Without calcium ion, your muscle cannot contract. Now, mitochondria is needed. There also, obviously, you see mitochondria all over here. These are mitochondria. These are mitochondria. And obviously, they are there to supply ATP. Muscle is going to contract. For this actin and myosin filament to contract, they are protein. They do the job. They are the machine. But for them to do the job, they need ATP to do their job, right? Okay, let's go. Now, when you look at this, you see here filaments. I told you earlier that you have the thick filament and the thin filament. And I told you that you have actin, which makes the thick filament, the thin filament, and myosin, which makes the thin filament. So actin makes the thin filament, myosin makes the thick filament. Again, actin, think about it, is thin filament. Myosin, thick filament. Now, so here is what the sarcoplasmic reticulum looks like. Here's the sarcoplasmic reticulum, but here is a terminal cistern, and here is another terminal cistern, and here is a transverse tubule. These three together makes what we call three, which is triad. All right, these three makes what we call triad. Now, when you want to study this, first thing I want you to look at this image here, and I will discuss some of the few things that are very, very important for us to look at on this chapter. I'm going to start from here. What you are seeing here is a thin filament. And the thin filament, like I told you, is made predominantly by actin filament or actin protein that is arranged in that appearance. That is the thin filament. Now, when you see this brown are actually the actin filament. You see, those are the actin filament there. Those are the actin filaments. Now, you will notice that there are some strands on it. This strand is called tropomyosin, the chain, and this lock here is troponin. Now, there are two categories of proteins that are very necessary for this process. The first category are I'm going to type now. The first category are the contractile proteins. The contractile proteins. And we call those actin and myosin. The second category of protein, we call them regulatory proteins. And those proteins are troponin and tropomyosin. Those are the two categories of proteins that we will be talking about: the troponin and tropomyosin and the contractile protein. All right. So the troponin and the tropomyosin are the regulatory protein. If we say a protein is contractile protein, what does that mean? It means those are the proteins that are necessary for contraction to happen. If we say that these proteins are regulatory proteins, it means these are the proteins that help to regulate contraction. So once I'm done with the anatomy now, I will take a pause, then I will record the second part, which will be part two, so that it doesn't get too long, to discuss the physiology of this process. Now, so the filaments are arranged. Now, when you see this, so the blue or the green cord you see around there, I call it chain. This chain is tropomyosin. Then there's a lock on it. The lock on the chain that you use to lock the chain is called troponin. Now, so the troponin, the tropomyosin, and the actin filament makes what we call thin filament. Now, so the red here now is a thin filament, and the purple is a myosin. Now, you notice that myosin have this projection. We call it myosin head. You see that? Those little projections, we call it myosin head. Now, the head of the myosin has to bind to somewhere which is called the active site on actin. But there are regulatory proteins there that prevent it from binding. And so you see tropomyosin binding on that same active site and troponin locking it up there. So they are not going to let myosin head fit into the active site. And if that doesn't happen, there won't be contraction. I'll be talking about that later. All right. So here is actin and here is myosin. You see this point here, we call this a Z line because it's zigzag, right? We call it Z line. This Z line and this is another Z line. Each Z line, from the beginning of one Z line to another, is what we call sarcomere. And sarcomere simply means a functional unit of a skeletal system. Just the same way a cell is a functional unit of a living thing. So the same way sarcomere is a functional unit of a skeletal muscle. Now the thick and thin filament, we've talked about that. You see there's an area where they overlap on each other here. All right. Now, the area when they overlap on each other is what we actually see and we call it striation. So when you say skeletal muscles have striation, it's simply the arrangement of the active actin and myosin protein. All right. So the point here we call this the actin again is what makes what we call the light band. So this is going to be the light band or I band, and then the myosin we call it dark or A band. So it's the same thing. All right, let's go. Now, contractile protein, I've talked about that. They are proteins that regulate contraction, which are the actin and myosin, right? Again, actin is a thin filament. Myosin is a thick filament. Actin makes the I band. Myosin makes the A band, which is the dark band. Actin makes the light band. All right. Then we have the regulatory proteins, which is what decide if there's going to be contraction or not. And that is tropomyosin and troponin. All right. Now, the filament. Myosin again, once I'm done with the anatomy, I'll pause, then I'll continue on the next chapter. On the next path, the video. Now, myosin is a protein, contractile protein, and again, I told you it's a thick filament. I told you that it has a head, and the head is what is going to fit into. These are the heads, right? The head will fit into the active site on myosin, on actin. This is the head. This head fits into what we call active site on actin. When the head fits into it, we say that a bridge has been formed. That bridge is called cross bridge. When cross bridge form, that is actually the genesis of contraction, which I will be talking about later. Now, thin filament, like I told you, is made up of actin, it's made up of troponin and tropomyosin. Those are the components of the thin filament, which is what you see here. Now, in relaxed muscle, the myosin binding site on each actin molecule is covered by tropomyosin, which in turn is attached to troponin. Does that make sense? So you see, there are tropomyosin that covers this, and I call troponin a lock, is a lock on the chain. So I say tropomyosin is a chain, and I say troponin is a lock. All right, so that will lock up the chain to prevent the chain from falling off. Now, if there's going to be contraction, I want you to start thinking now, since I'll be talking about that later. If there's going to be contraction, that means something must unlock troponin. For contraction to happen, troponin must be unlocked. So what unlocks troponin? I'll be talking about that in the next video.